Hi, everyone. Today, our goal is to talk about tracking inheritance, and we're going to see a couple different ways we can do that. The main one that we're going to be working with is pedigrees. So pedigrees can be used both by people for like family inheritance, but pedigrees are also used for breeders, for example, if they want to know what is sort of the family history of some trait within some animal that they're trying to breed. So very, very useful tool. We're going to see, you're going to see at the end, there's some practice also that I've attached for you. Essentially, what the pedigree does, though, is look at more than one generation. So we're not simply doing a Punnett square where it's just the parents and just their offspring. Now we're often looking at more than two generations. So some pedigrees can be quite large, looking at many generations. But the goal is that we're not making predictions anymore. We're now looking at what actually is the trait. So I'm going to walk you through some examples of that. We're going to have some practice to make some pedigrees in here as well. So here is just an example of the symbols on the pedigree. We have squares or circles representing males or females. So with a square, that is a male. You'll notice that it says normal, so this would be not having the particular trait. And then a shaded one is actually having it or being affected by the trait. Likewise with females, circle, and then without shading versus with shading. So just want to mention, we don't only use this for things that are diseases or disorders, although that is what we tend to use them for, because usually many of the traits we don't really care that much about, but if it's something that has to do with a genetic disorder, it'd be really useful for us to show that on a pedigree. We're also going to talk about on the second page that some disorders, diseases, even just traits themselves, it does depend on the biological gender of the person. So we're going to look at that on the second page. Just to give a little quick introduction, what I'm showing on the side in blue are Roman numerals. Those are showing generations. So one being the earliest generation, and then three being the most recent generation. So in this particular pedigree, we have up here the grandparents, their children, and then the current generation or the most recent, which would be like the grandchildren. So it's like showing a whole family. The Arabic numbers, which are just regular numbers, 1, 2, or 1, 2, 3, 4, and so forth, those are the general birth order. So why we use these numbers is it gives sort of like an address to describe each of the people. So if, for example, I wanted to talk about this person at the top that's shaded in black, I would say Roman numeral 1-1, and that refers to this person. Now, I had shown it above. We have a symbol for mating. So this line that goes between two people left to right, that is showing a cross between two people. Lines that go downwards, those are showing their offspring, okay? So again, here we have like a little legend, which is like affected versus unaffected. And we will see really soon why that's useful. I'll just show in the second generation. I was saying about the birth order. So we had like the first sibling, the second sibling, the third sibling. Number 2-4 is not a sibling. They don't come from the same parents. They are the mating partner of person 2-3. So this person, number 2-4, they are not necessarily written in the order that they're born. They're not necessarily the youngest person in this particular generation. But that's just the easiest way for us to draw it. So the birth order, it applies to people who are actually born into that family, but any mating pairs, they are just put in after the fact. So another thing I was just mentioning was like, why is this useful? So many genetic counselors, they will use this with families who think that there is a chance that their child might inherit a genetic condition. So just like with other traits, it's very possible that a genetic condition gets hidden in one generation, could even be hidden in a couple of generations. So it could skip like more than one generation, and then we see it resurface later on. So genetic counselors you would use if you are concerned about that happening. You yourself might worry that you have a genetic condition. You would also use a genetic counselor for that. So that's just like a career thing that you can think about. Down below, I've done an example of creating a pedigree. Again, this particular one is nothing that's, let's call it bad. It's just any physical trait that we wish to do a pedigree for. So in this trait, I'm describing a family, which is the brown-eyed woman whose father had blue eyes and whose mother had brown eyes, marries a brown-eyed man whose parents were also brown-eyed. They have a son who is blue-eyed. So draw a pedigree, assuming that brown eyes are dominant to blue eyes. So that last phrase that I've underlined in red, it's actually really important. So what I've made it is that affected is the brown eyes, the dominant trait. So I've drawn in green on the side here is just like my little legend, so to speak. But also I've put in somewhat of the let statements, right? What am I representing each of those traits by? So just as like a brief reminder from Friday, because this is kind of a big concept, the capital B and small b, those represent alleles. Those represent options that the eye color can have. Now, I've talked about this before. With human genetics, we don't only have those two outcomes. So this is definitely an oversimplification. There are sort of other phenotypes, other appearances that happen in between. We won't be seeing anything crazy like that in our pedigrees. We're going to keep it really, really simple. So you'll see that as we go through. So basically, what I start doing is pulling information from this statement or from this little question. So it really depends where you want to start with this. I might actually choose to start with the grandchild or the most recent person just because his is very simple. They have a son who is blue-eyed. Okay, so again, I've said that blue eyes are recessive, right? Brown eyes dominant to blue eyes. So I would almost start my pedigree by doing this person first. He's the only person in the third generation that we know about. So I would draw him in using a square with an open box, so not shaded because that's the recessive trait. So then I start working backwards. What about his parents? So his mother is brown-eyed and his father is also brown-eyed. So I symbolize that by doing shaded square and shaded circle. Again, the birth order here, we cannot be sure because they're not siblings, obviously, but we just can put them whatever order and that's fine. And then I work backwards. So the reason I've underlined for the woman, her parents, because the father has two parents that are brown eyed. So that's fairly simple. We already know, okay, they have brown eyes. But the mother, it's very interesting. She has a father who has blue eyes, a mother with brown eyes. So we suddenly have up here is an unshaded box. A grandfather that has blue eyes. So what I was saying before, we can actually visually see on this diagram, on this pedigree, that the blue eye trait has skipped a generation. Okay, it's hidden in the parents. Now, additional to this question, which I did not ask you, but I think it's a useful thing to sort of think about, is how do the Punnett squares work out for this? Because on Friday we worked on Punnett squares, so we're just sort of like bringing that same concept through. If I said that blue eyes was recessive, every person who has blue eyes in here must have the genotype of small b, right? That's called homozygous recessive, two of the recessive allele. So I've written that in just above or below, depends where I had space, and then put BB there. Now, the other people in here, they all have brown eyes, right? And we said on Friday that there's options that it could be two capital Bs or it could be heterozygous, capital B, small b. So for each person, we need to think about what makes sense for them to have. So in the second generation, for example, I've written in that it has capital B, small b for both people. The reason why this works out, if you do draw out a Punnett square, they cannot be homozygous dominant because they end up with a son who is, of course, the recessive phenotype, recessive genotype. So each of these parents in generation two, each of them must give one small allele, one recessive allele for the blue eyes. So that's how I know for sure that's those parents. In generation one, you're going to see that the people I'm not sure about, I have put like a B and a little underscore, or B and a blank really is what it is. So B and blank means that I cannot be sure which of these parents has which genotype. I see that they have brown eyes, so I know that they for sure have the capital B or the dominant allele, but the little space beside it is because I don't know for sure, is it two capital Bs or is it heterozygous? I know that for sure one of the parents has to be heterozygous in the first couple in 1, 1, and 1, 2, because again, their child ends up with a recessive allele somehow. But I don't know which one is which. So I leave it as B with a blank. Okay. If you look online or if you like start Googling different examples, you will see that they denote it in different ways. This is how I will denote it. So I would encourage you to do that too. Some people will put it as like the two options that exist. So they'll write both options there. That's acceptable as well. I just feel that this indicates that we aren't sure. We can't be sure of it. Okay. And the same reason for person 1-4. It's possible that they do have a small case B here. If you draw out the Punnett square, that scenario would still produce 50% of the person down here, this 2-2 person. So if it's helpful for you, you can always draw out Punnett squares of these scenarios when you're trying to predict the genotypes. Now, on the second page, this is where it gets really interesting. And I had a question about this on the chat last week, which was, does gender have anything to do with this? And the traits that we've done thus far and what was on the problem set, those ones did not have anything to do with biological gender. So it was didn't matter if it was a male or a female. Sex-linked inheritance is dependent on the sex chromosomes, on either the X or the Y chromosome. So just as a quick reminder, two X's would be biological female, XY would be biological male. We're not going to look at things like Turner syndrome and Klinefelter syndrome for pedigrees. That was to remind you they would have the extra Y chromosome, or excuse me, extra X chromosome for Klinefelter and one fewer X chromosome for Turner syndrome. So we're not going to look at those examples. We're going to assume that it's only two, right? Assume that it's a typical chromosome number. If the allele is found elsewhere, so not the sex chromosomes, we call that autosomal control, okay, or an autosomal dominance pattern, and I'll show you some examples of that soon. So as I was just mentioning, we have just the two sex chromosomes, X or Y. So two, we have X-linked or Y-linked disorders. And it's not always like a disease. It could also be just a trait that happens. So one example is like male pattern baldness. So baldness is not some type of like scary disease. It's just a pattern that passes on. Same thing with like color blindness. So again, just going back to what I said earlier, it's not necessarily like some scary disease. It could be just a trait that follows that pattern. So the two patterns then are X-linked or Y-linked. More interesting to us is going to be the X-linked, and the reason is because it affects far more people. Remembering that females have two X's and males have one X. Y-linked only applies to males, of course, because they have a Y chromosome. Females do not have a Y chromosome. So what we see with X-linked is that there is a trait that is on specifically the X chromosome. So I mentioned those examples earlier, color blindness, male pattern baldness, also hemophilia. And hemophilia we are going to talk about a little bit more later on. That one is a very serious disorder, could be life-threatening, but there is treatment for it now. We will talk about it later. So here with my sort of like, how do I denote it? So I have X with a capital R in the corner, which is a superscript. This represents if the X-linked trait is dominant, or I could have an X-linked recessive trait, which is the trait is recessive. So X with a superscript of a small r. So depending on what the disorder is or what the trait is, we're going to denote it differently. So with here, we're always using X as the base. So we want to show whoever is looking at it, remembering our let statement concept. We want to show who's ever looking at it that it is on the X chromosome specifically. We don't need to do that with any of the autosomal traits because they don't depend on gender, but here it will depend. So I'll show you an example just below of hemophilia for that. The Y-linked is that there's the trait will end up on the Y chromosome. There are very few examples of this. One example is reduced fertility for males. And because females don't have the Y chromosome, only males are affected by these disorders. So we're going to see on some pedigrees later on that when we look across the pedigree, we can see very, very quickly if it's X or Y linked, or if it has nothing to do with X or Y. So we'll talk through that on the next sheet. Down below here is an example of sex-linked inheritance. And so we have a problem, just like we would have the Punnett square problems before. So hemophilia is an X-linked recessive disorder. That first statement or that first line is extremely important. That allows us to write our let statement. So we're told it's X-linked recessive. So I've made my let statement, let X with the small r for recessive, right, represent hemophilia. X with the capital R superscript will represent an unaffected individual, okay, so that doesn't have hemophilia. So what I've done down below here is list out the parents, okay? So I've said predict the probable outcomes for the cross or the mating between the mother who is a carrier of hemophilia and a father who does not have hemophilia. So I want to mention that carrier word. The carrier means that she herself is not affected by the disorder, but she carries a gene for it. So, what that translates to essentially is that she has to be heterozygous. She has to have one of each of the alleles. So, if we write out her genotype, we have X with a capital R and X with a small case r. So, she carries that recessive hemophilia allele. But just like with complete dominance, because technically this is complete dominance, the capital R allele overshadows it or dominates over it. So that's not what we see in her phenotype. So she is the carrier. Now, the father is unaffected. So he has an X and a Y chromosome. His X chromosome will have the unaffected allele, which is capital R. His Y chromosome is a totally different chromosome. So you'll notice he has nothing written on the Y chromosome. Reason being is that that trait does not exist on the Y chromosome. So we just leave it blank. We don't write anything on the Y. It is important we show the Y because when we're actually going through the Punnett square examples, or if we're drawing pedigree for this, we do want to see if it's a male or a female, right, that gets affected by the disorder. So what I've done down on the side here is then take these two parents and cross them together because what we're looking for is the probable outcomes. So if you look at my Punnett square, and I know it's a little bit small here, you'll have the cross between male and female that we've listed. And so when I cross those, I see in the top row, I have females. Both of these females are unaffected because they both have present that dominant allele of X capital R. So those females are unaffected. If we look at the males, I find one affected, which is the X small case RY, and I find one unaffected. So there's a few things that we could list here. We weren't really asked anything specifically, so I just left it as blank. You could be asked what percentage of the females are carriers, or what percentage of all children actually have the disease, or so forth. So you're using your Punnett square to take away that information. So that's our sex-linked inheritance. I wanted to show this actually on some pedigrees now because, like I said, part of what is so useful about the pedigree is that we actually see genders each generation, and we can see that traits can jump generations and so forth. So I'm going to give the example here of color blindness, okay? And I'm going to walk you through how I've analyzed this pedigree. So I'm going to first actually zoom out a little bit here. So this one is already a pre-drawn one. And so before I even get into the questions that are on the side here, I can see very, very clearly that there is a discrepancy, there is a difference between people who have the disorder and people who don't. So in this case, the color blindness is going to be the shaded ones. I've made a little legend down below here. The unshaded means normal vision, okay? So my first glance at this, because what these questions will ask is what type of inheritance pattern is displayed. Okay, we're going to see in the examples several different types. So you all follow through with what are the different patterns that could happen. But as I mentioned above, it's either X-linked or sex-linked, I should say, or is autosomal. So it's one of those two options. Within sex-linked, we could also have the X-linked or we could have the Y-linked. So we'll go into how we know that. So at first glance over here, you should notice that there are only males who have the disorder. So that's the first thing that when I look at this, I'm like, wow, that's really obvious to me. So in terms of the pattern, we are predicting what the pattern is. We don't know it for sure. Okay, so without more information, we really can't know for sure. We're making our best prediction based on the information that's given to us. So in this example, we see only males, and that immediately tells me this is probably sex-linked. So that's going to be my first step that I've written in green here. Then I'm guessing it is X-linked recessive, and I'm going to walk you through how I think it's that as opposed to Y-linked, because as I said, only males have it, okay? So down below here, you'll see my explanation, so to speak, of why it's that way. So it has to be recessive, number one, because if not, we would see that these fathers that are shaded in black, they would for sure be passing that trait on to their daughters. Because remembering, and I'll show it in the Punnett square, remembering that males, they always pass their X chromosome onto their female offspring, onto their daughters. But in our actual pedigree, we don't see that as the case, okay? So if it was X-linked dominant, we would see that also females have it, but that's not the case in our diagram. Now, I just want to mention down below, I've put like a little blue Punnett square. The reason why I've put this here is just to show you what the possible scenarios are. So what is written here in blue, this represents what the actual Punnett squares would look like, okay? What I've written in red here, a small case r, this is to prove to you that this is not a Y-linked trait. So I'm going to refer now to individual 2-3. Okay, individual 2-3 is affected with color blindness, but if you look at his offspring, which are 3-3 and 3-4, his son does not end up with the disorder, right? His son has normal vision. So going back to why I've drawn this with the small red R, it's to show what is not the case, what is not happening here. So this small case r is not true, and I'm just proving it to you why it is not Y-linked. Because if it were to be Y-linked, it would be passed from every father to every son, and that's not the scenario that we see in this example. So the trait that's dominant here is normal vision. Part of why I know that is that I can look across this entire thing and I see that very few people actually have the color blindness. So I can make that prediction. Again, for reasons I just mentioned to you, when I'm tracing it generation to generation, if I did a Punnett square like I've done in blue at the bottom, I can test out, does it make sense for this trait to be dominant or recessive? And you will quickly find that it only makes sense if this trait is dominant. Number C is asking, how does individual 4-1 have color blindness even though neither parent has it? And if you look at this situation down here that I've circled in, or excuse me, blocked in red, this is a trait that was hidden in the parents, but reappears in the next generation, just like we talked about in Mendelian genetics. So the mother that is 3-2 here, she must be a carrier of the disorder, okay, a carrier of color blindness. And it doesn't appear in her because, like we said, color blindness is recessive, okay? So hopefully that helps explain. And I get the question a lot, how do you know this for sure? The truth is we don't, okay? We are just making a prediction based on what is really only a small amount of information. It's really only 4 generations. If we wanted to prove this further, we need to look at way more generations, okay? But we are making our best prediction just based on the pictures that we have. So going on to a couple of other sort of types of patterns here. I have genotypes, so fur color in mice. You're going to be looking at that in the lab also, but that's sort of a separate issue. That one is looking at dihybrid crosses, so more than one trait. But here we're going to talk a little bit about fur color. Now in this particular pedigree, and I know I've written on top of this one, but I've already given you what the shaded and unshaded means. So the white fur versus the black fur, female versus male. Now, first question with these is usually what type of inheritance pattern are we showing here? So my initial analysis, again, as I said before, I look across the entire thing and I see, is there a huge discrepancy between males and females that have this trait? And when I look across, I really don't see that here. So immediately that signals to me this is probably going to be autosomal as opposed to sex linked. So I've written here it is probably autosomal. Okay. So autosomal inheritance pattern, we're not using the X and Y chromosomes to denote anything because this trait is not on the X or Y chromosomes. Again, we're making this prediction. It could be that we're wrong because we're only looking at 3 generations, but we always choose what makes the most sense based on the information given. So I mentioned which fur color is dominant. And I've said here black is dominant to white. And this is because if we actually follow what's happening here, very similar to what Mendel did, we can see that some traits hidden in the first generation reappear in generation 2. So I'm going to refer to the green box that I've drawn around this family on this side, on the left side. So here, generation 1, I have 2 individuals that both have black fur. In the next generation, I look at their children, and there's a 3-to-1 ratio of individuals with black fur to individuals with white fur. So this white fur trait must have been hidden in the parents. So what I've done down below, which is this green Punnett square, is just to prove how that must be the case. Okay. Now we're going to see it doesn't always end up in the predictable ratio, and hopefully you notice that with the problem set, it's not always going to be exactly the amount that you think, okay? Because as I've said before, each individual offspring is basically a reset of the Punnett square. So if I have, for example, 4 children, it's not necessarily going to be always 2 boys and 2 girls, okay? Just because that's the Punnett square, that's what the Punnett square predicts. Each and every offspring is basically a reset of the probabilities within the Punnett square. So what I'm doing essentially in part C is then predicting the genotypes, which I explained to you with the blue and brown eye color as well. So I'm going to start off with those who are recessive because, as we said before, that's always the easiest. We know them for sure because they don't have that dominant trait. I've written down here my let statement for this, capital B for black fur, small b for white fur. So immediately anyone that is that has white fur, I label them as small b, and I kind of fill in the whole Punnett square with that first. Then I start to look at those who might be heterozygous. So again, for the offspring to have any white mice, we know that it has to be heterozygous because otherwise it's 2 dominant alleles. We can't get a white allele from that. So I'm trying to show that to you in these Punnett squares down at the bottom. So again, this red box that I've kind of boxed in, this family, I'm basically showing to you why it has to be the genotypes that I've listed here. And likewise, if we follow down to the 3rd generation, individual 3-4, they have to be heterozygous because one parent gave a small b, one parent gave a large b. So we essentially continue looking through this, and I mentioned down here, for heterozygous, we can predict that very accurately by looking at the offspring. Now I want to just go up to generation 2 and the people 1 to 3 that are up here, or the mice rather, why I've put B with the little dash or B with the space. To repeat myself again, these ones here, I cannot be sure whether they are 2 capital B's, which is homozygous dominant, or whether they're heterozygous. The only way for me to know that is that I would have to breed them again. I would have to cross them with other individuals and look at their offspring. So without looking at their offspring, I cannot say for sure what their genotypes are. And that happens often when you have a small Punnett square. So that's just something to keep in mind as well. We're going to go on to another inheritance pattern, which is also autosomal. So down below here, again, the question whether it's autosomal or sex-linked. So in this one, it's autosomal, and I know because when I look across it, I see actually that the male and female division, it's pretty equal, so I don't immediately assume, oh, this must be sex-linked. So then I have predicted the genotypes of each individual in the pedigree. So I've said here, I'm going to make capital B the normal or the straight thumb. So I have straight thumbs. If you flex your thumb and it bends backwards, like sort of gets curved, that would be a hitchhiker's thumb. So it's a really common trait actually. I see in here that actually most people have, or there's sort of a mix. We have people that have normal straight thumbs or not. Now I've mentioned down here in blue, I'm just going to zoom out for a quick sec. The trait is not present in parents in the second generation. So if you notice these two parents in the generation 2, they do not have the trait, but suddenly in their offspring it appears. Okay, so this tells us that hitchhiker's thumb is recessive because it wasn't present in here, then it reappears in the third generation. So I have used that information to make my sort of let statements on the side. Then when I go to actually put in the genotypes, I'm going to use the same strategy like before. Anyone who has hitchhiker's thumb, the shaded one, I put in 2 small b's. That one I know for sure. And then I follow suit doing the same pattern with all of these. Now, if you find it easier, which some people do, you can always draw yourself like a small Punnett square off to the side, which is basically to say, okay, what is the actual possibilities that can come of this? I'll just reiterate and mention again, it's not always equal to what you predict in your Punnett square. So your Punnett square might predict like a 50-50 split, but just like with if I had 4 children, I could end up with all 4 are girls or all 4 are boys, right? It doesn't change the fact that each one of those children had a 50-50 chance to be male or female. So just something to keep in mind while you're working on these. The last page in here, and I know there's like some practice for you guys to do that's linked to the lesson, but the last one that I'm going to do in here, another sort of like useful thing that we use tracking for, is tracking inheritance over generations, which is not necessarily using a Punnett square, but we can just use like almost descriptions. So an example, a cross between 2 chinchillas. There are 2,020 gray chinchillas and 675 white chinchillas produced. Determine the probable genotypes of the parents, which allele is dominant, and assume the trait is influenced by only 2 alleles and follows Mendel's inheritance. So I'm going to use capital R and small r to represent the alleles. So based on the description, I see that there are lots of gray and there are only a couple of white. So immediately I start to think, well, gray is probably dominant. The other thing that you can do is actually think of the percentages. So if we think of the total of this, which is 2,675, 95 actually, if I add these two together, then I will find that it is about a 3-to-1 ratio. Okay, so for every 3 gray chinchillas, there's approximately 1 white chinchilla. So down here, I've sort of shown what the math of that looks like. Okay, so that tells me that the capital R, or the dominant, will be the gray fur color and small r will be the white fur color. So when I work backwards, okay, I look at what the percentages actually are. This should say 2695. Pardon me, I miswrote it, and this one is written correctly. But what we're looking at is the percentages. So like I mentioned, 2020 out of 2695 is about 75%. And then 675 out of 2695 is about 25%. So these are not exactly equal to the Punnett square, but they're pretty close, okay, because we've got lots of individuals. So what I've drawn here in the black is I'm basically playing around with what makes sense for the genotype of those parents to be. So the ones that produced these siblings, right, 2020 and 675. So most likely, both parents are actually heterozygous. Okay, now I mentioned here the actual outcome, which is the numbers 2020 and 675, is not exactly equal to the prediction. Just like in a family, if I look at the offspring, it's not necessarily 50-50 of males or females. Okay, thinking about larger sample sizes. The larger your sample size gets, the more offspring you have, the closer you will get to the predicted outcome. So as an example, if I look at my family versus I look at the entire population of Ontario, I will see that Ontario is very much closer to a 50-50 male and female as opposed to my family, which might be 100% females. Okay, So just something to keep in mind, as sample size grows larger, you get closer to the predicted outcome from your Punnett square. The last one I'll show you in this lesson, which is kind of fun, are called test crosses. And test crosses we use if we're unsure of a parent's genotype. So as mentioned previously with Mendel, for those who have a dominant phenotype, we don't know for sure if their genotype is heterozygous or homozygous dominant. So in order to determine this, we will take a parent with an unknown genotype whose phenotype we can see is dominant, and we will cross it with a homozygous recessive individual. The reason we do this is that the two small case Bs, we know for sure what their genotype is, because if they had a capital allele in there, a dominant allele, it would give us the same problem once again. We wouldn't be sure what their genotype was. So based on the offspring of that test cross, we will then make a prediction, okay, which of these made sense for the other parent to be? So I'll walk you through an example of that. I think these ones are kind of fun. So here we have rabbits, long ears dominant to short ears. So that's fine. We can make our let statements. Capital E represent long ears, small e represent short ears. So it says a test cross is performed on a rabbit with long ears. Okay. Immediately when I see test cross, okay, that tells me I have to be crossing it with a parent who is homozygous recessive, okay, who has that recessive phenotype. The rabbit with the long ears, I am unsure of. I don't know if it's a capital E, homozygous dominant, or heterozygous, large case E and small case E. So essentially what I'm doing below is showing what the two options are. I've mentioned right here that it results in 3 rabbits with long ears and 1 rabbit with short ears. So that is the offspring of that cross. So it says determine the genotype of the long-eared parent. So like I wrote up here, is it EE capitals or is it heterozygous, E with a small e? So here's my 2 options. And you'll notice that in both options, I've used the test cross. I've used a homozygous recessive. So the small case E's on the side. Option 1 is if the first parent that was unknown is 2 capital E's. And so if I notice, 100% all have long ears. In my actual outcome that I was told, though, there are some that have short ears. So I know option 1 is not possible. Option 2, though, where I have the heterozygous parent against the homozygous, I see that actually there is a prediction of 50% heterozygous, which would have long ears, 50% homozygous recessive, which would have short ears. Okay. Now I want to mention on the side, I'm just reiterating again what I said earlier. It is not the exact outcome that was observed. Okay. So the observed outcome was actually 3-1. But this is a possibility. So if I allowed these rabbits to continue breeding and I made, let's say, 100 rabbits, I would get closer to the 50-50 outcome, right? However, I only have information about 4 individuals here. This one, option 2, is the only one that makes sense, because if it was option 1, none of them could wind up being short-eared. So my sort of like therefore statement, the genotype of E with a small e or the heterozygous is the only one possible for this unknown parent. Otherwise, there's no possibility of us getting any individuals with short ears. So I encourage you to try some more examples of pedigrees. I do find that this is one that people get a little bit confused on because there's a lot of different examples possible. So please do try out some examples. Feel free to come by in office hours as well. I'm happy to go through a few more examples of these.